I want to welcome everyone to the NIH Director's Wednesday afternoon lecture series. My name is Gigi Stortz, and I'm a senior investigator in the Child Health Institute. It is my great honor and pleasure to introduce my good friend, Dr. Rachel Green, who will be presenting a very special lecture, the 10th Annual Marshall Nurnberg Lecture, where we salute one of NIH's great scientists. Now, the NIH director, Dr. Francis Collins, very much wanted to be with us today and to do this introduction, but earlier today he was called to the White House for an important meeting on coronavirus testing. He wanted me to say that he sends his deep regrets. He said that Marshall Nurnberg holds a special place in his heart. So Dr. Nurnberg, who is shown on this picture, was one of the stars at NIH. Dr. Nurnberg conducted the groundbreaking research that led to the unraveling of the genetic code. In 1958, he began investigating the relationship of DNA and protein production via the hypothesized RNA intermediate, messenger RNA. He and his colleagues demonstrated that a synthetic RNA could serve this function and laid the basis for much of our understanding of protein synthesis from amino acids. He then solved the genetic code, starting with the famous discovery that PolyU, you can see U are great, PolyU messenger RNA encodes polyphenylalanine. He received the 1968 Nobel Prize in Physiology or Medicine for the interpretation of the genetic code and its function in protein synthesis. He was the first NIH scientist and first federal employee to win a Nobel Prize. After this work, Dr. Nurnberg focused on neurobiology, neuroblastoma cells, and the relationship between homeobox genes and neuronal development. The Marshall W. Nurnberg Lecture was created to celebrate this amazing legacy, and you can see the amazing people who have been awarded this honor. So now let me introduce Dr. Rachel Green. Rachel is a Bloomberg Distinguished Professor of Molecular Biology and Genetics at the Johns Hopkins University School of Medicine. She's also a Howard Hughes Medical Institute investigator and director of Hopkins Biochemistry, Cellular and Molecular Biology program. Rachel received her undergraduate degree in chemistry from the University of Michigan, and then a PhD in biological chemistry from Harvard University under the mentorship of Jack Szostak. After postdoctoral training at the University of California, Santa Cruz under Harry Noller, Rachel moved to Johns Hopkins in 1998, where she has been ever since. It is in Dr. Noller's lab, I think, where Rachel fell in love with ribosomes. Her research on the amazing ribosome, which combines biochemistry, genetic approaches, and ribosome profiling in bacteria, yeast, and human cells, has revealed fundamental mechanisms of protein synthesis. These studies ultimately are providing insights into a wide range of human disorders that disrupt ribosome function or the cellular responses to ribosome dysfunction. Some of Rachel's more recent work, which I think will be the focus of our talk today, has been on ribosome-mediated quality control pathways that are triggered on difficult to translate mRNA sequences deriving from genetic or environmental insults. She has found that such translational distress signaled by colliding ribosomes leads not only to mRNA-specific quality control events, but also to the activation of cell-wide signaling and transcriptional responses. Rachel has received many honors, including a Burroughs Wellcome Career Award and a Searle Scholarship Award earlier in her career, and more recently election to the National Academy of Science, National Academy of Medicine, and the American Academy of Arts and Sciences. I personally want to mention that on top of these accomplishments, Rachel is a great cook and the mother of 3 beautiful and talented young adult children. The title of Rachel's talk today, as you can see, Is colliding ribosomes function as a sentinel for cellular distress? Thank you for joining us today, Rachel.
Thank you, Gigi, for that lovely introduction. We are good friends indeed. So I couldn't be happier to give this lecture. We all have our favorite science stories, and while the discovery of the double helix was a good one, mine has always been the discovery of the genetic code, probably for obvious reasons. I was a chemistry major in Ann Arbor, where I noticed Marshall Nurnberg did his PhD work, and I remember the class where I learned about how his chemical and biochemical insights led to the unveiling of the code. It really must have been an amazing time to be a scientist, and I'm really honored to be here to reflect on this. I also want to give a special nod to my colleagues at the NIH and thank them for inviting me and hosting my visit. I've had a great day of conversations, and I would add that building on its earliest history, the NIH remains a very special place for those with interests in the ribosome. So with that, I'll get started. As Gigi mentioned, this is my title, and what I'm going to tell you about is how we think ribosomes play a surprising and central role in sensing cellular distress. These are my conflicts of interest, which were already mentioned. So here we go. So to set the stage, what I'm showing you here is a cryo-EM tomography image of a yeast cytoplasm from Liam Holt's lab. And what's shown in gray are the organelles, and what's shown in aqua are the ribosomes, measured in this study at 23 micromolar in concentration and 20% of the volume of the cell. And so I've highlighted that what I'm going to tell you about is how ribosomes could be a fundamental signaling molecule. And I think looking at this view of the cell, it doesn't seem like such a preposterous idea. So with that in mind, what I'm outlining here is what my lab and others have studied for a very long time, which is how it is that ribosomes in eukaryotic or bacterial cells get on at the 5' end of messenger RNAs. They scan to find a start codon in eukaryotes. The large subunit joins, they elongate through the open reading frame to reach the stop codon, and the messenger RNA is polyadenylated. And like traffic on the road in the morning when we used to commute to work, ribosomes move most effectively when there are no sort of collisions or slowdowns. So that's depicted here. As Gigi mentioned, what we've been interested in for a number of years are a collection of quality control pathways that are important in eukaryotic cells for identifying problematic messenger RNA species. So these problems could be encoded, genetic errors or processing errors, or they might be environmental, as you'll see. And what we've known from some time is that when the ribosomes encounter problematic sequences in the messenger RNAs, there's a whole collection of quality control events that are triggered, akin to a car with a flat tire being pulled off the road. What we think the most important task of these quality control systems is to deal with the incomplete nascent peptides that are generated on such messenger RNAs, as these can be proteotoxic. So that's probably the primary event that takes place. We know that in yeast, mRNA degradation is tightly coupled to this nascent peptide degradation, and we began our interest in this area focused on the idea that ribosomes had to get off these problematic sequences. So for a number of years, we and others were interested in understanding how a slow ribosome in the cell might signal a problem. And really great insight came to this problem a number of years ago when Hanny Zayer at Washington University found through biochemical experiments that really involved varying ribosome load on messenger RNAs through manipulation of the 5' UTR. What Hanny found was that it wasn't in fact a slow ribosome that was the trigger, but it was such a slow ribosome that it led to a collision. This collision presented a new interface that didn't previously exist. And this interface is able to recruit an E3 ligase that ubiquitinates small subunit proteins of the ribosome to target all of these downstream events. So we and others have been excited about the idea that collisions are important for these pathways. But what I'm going to tell you about today is what we think happens when these pathways are overwhelmed, for example, by broad environmental distress. What we think happens when that takes place is that instead of a simple fix of pulling a car off to the side of the road, when you have a bigger problem, what happens is the cell activates general stress responses, in this case referred to as the ribotoxic stress response. And we think the colliding ribosome is central to that.
Rachel, real
yes.
quick before you get into it, you have some colored pencils on your screen. I don't know if you can just click that closed.
How is that possible?
If not, it's not major.
Okay, I'm good. Oh, how is those are visible? That's so weird. Anything else?
Oh.
That's great.
Perfect.
Thank you, Gigi.
Sure.
Just keep me posted. Okay, so what I'm mostly going to show you in this talk is cartoons, but this is to give you a sense of what this colliding ribosome actually looks like. You know, Hanny did this beautiful biochemistry, then the cryo-EM people come along and they give us a beautiful image and they get all the credit. But these are beautiful images from Roland Beckman and Manu Hegda's lab of colliding ribosomes. This is the lead ribosome. For a ribosome person, it's in a so-called classical state. The colliding ribosome is in a rotated state. There are a number of satisfying features about this structure. First of all, the ribosomal proteins that are ubiquitinated by that key E3 ligase are right here. RAC1 has long been implicated. This is a ribosomal protein that's long been implicated in being important for these pathways. So it's a beautiful structure that reveals a lot. I'd like to point out that the messenger RNA here that loops between the two ribosomes is actually inaccessible to RNases because the packing at this interface is so tight, and that will be a useful biochemical tool. So as you're looking at my cartoons, you should think about this. So how did we, next question I want to present is, how did we start thinking that collisions might be important for these pathways? And that really all grew out of some ribosome profiling experiments that Colin Wu, a faculty member at the NIH now, and a postdoc in my lab at the time, did using ribosome profiling in yeast. Ribosome profiling, as you all know, was developed in Jonathan Weissman's lab with his postdoc Nick Angolia, and it really has transformed the field. We've worked very hard at using it as sort of a tool for doing in vivo biochemistry to study ribosome function. The basic idea is that cells are lysed such that all the ribosomes are displayed along the messenger RNA, protecting a footprint. If you use a non-specific RNase to digest the free messenger RNA, what you yield is monosome ribosomes, ADS ribosomes, and inside each ribosome is a small mRNA footprint that can be cloned and sequenced and then aligned to the transcriptome to look at where all the ribosomes in the cell are sitting in condition A versus condition B. We've worked hard to sort of refine this protocol. The details are not important. We actually use two drugs to stop translation, cyclohexamide and tigecycline, which actually yields two footprint sizes, one 28 nucleotides in length and one 21, that correspond to different states of elongating ribosomes that provide us with really lots of beautiful information about translation. For the purposes of today, I won't focus further on that. To give you a feeling for what these data look like, what's shown here at the bottom is sort of a compilation of data where we've done a metagene analysis where all the start codons in the genome are aligned. And what you can see is there's very little ribosome density in the 5' UTR, a big peak at the start site, and beautiful 3 nucleotide periodicity of ribosomes reading along a message in aggregate. A similar analysis at stop codons reveals similar things, 3 nucleotide periodicity, a nice stop codon peak, and ribosomes getting off at the stop codon. So it's been a powerful tool for us to study all sorts of things. And one of the things we chose to study with it was cellular stresses. And this is all while we were in yeast. And Colin did an experiment where he exposed yeast to oxidative stress, and he made a number of interesting observations, 2 of which are outlined here. First, what Colin observed was that MAP kinase signaling pathways were activated under oxidative stress conditions, and that was long known, but our methods of looking at it using ribosome profiling were the first to do that. What Colin also observed was that, to our surprise, that certain codons were problematic under oxidative stress conditions. In particular, proline codons were problematic. We don't know why, but something was going on with proline metabolism. So if we do the same sort of metagene analysis and line up ribosomes at proline codons like CCG, you can see in unstressed cells, prolines are a little tough, but in oxidative stress conditions, they're really tough, as though there's a problem. And we further observed that if you look 10 amino acids back, which is exactly the distance from the A site of the lead ribosome to the A site of the lagging ribosome, we noticed another little peak, which suggested there might be collisions happening. And because we've been so focused on collisions in our other projects, focused on quality control, we began to wonder if this activation of the MAP kinase signaling pathways might derive from collisions at these problematic sites induced by oxidative stress. And so that really was the beginning of us thinking of this connection. So that was all Colin, as is the next story is going to be. Sorry, I jumped ahead here. What I'm going to tell you now are how that insight led us to identify 2 different proteins. One is ZAC, a MAP3K, The other is EDF1, a transcriptional coactivator, both of which we found bind to colliding ribosomes under stress to induce cell fate decisions. So as I said before, Colin was responsible for all of that yeast work, and Colin Wu, now at the NCI with his own lab, was responsible for all of the ZAC work that I'm going to discuss here. So with that yeast result in hand, we became interested, as I mentioned, in signaling pathways and how oxidative stress might generally lead to the activation of MAP kinase signaling, at the bottom of which is P38 and junk, two kinases. And so we began thinking about this, but oxidative stress is tough. Oxidative stress modifies lipids, proteins, RNAs, everything. So it's hard to get to mechanism from that for us. And in talking to our colleague downstairs, Sergi Rijot, we asked if there was sort of a more proximal stress that we could think about. And what Sergi said to us is, well, in fact, I study MAP kinases, and the best way to activate these pathways in mammalian cells is with drugs like anisomycin, cyclohexamide, and ricin. And it won't escape your attention that these actually are drugs, all of which are known to inhibit the ribosome translation elongation cycle. So this is a ribosome taking in a tRNA, making a peptide bond, and translocating. And these three drugs block each of these three different steps in the elongation cycle, suggesting that we could get much closer, that we had a ribosome-centric way of studying these pathways, and we could stop talking about oxidative stress. I should mention that this was a pathway previously discovered by Bruce Magan's lab 20 years ago. It was known as the ribotoxic stress response, and he certainly appreciated that these drugs targeted the ribosome and that the ribosome was at the heart of this stress. But for us, it meant something we could get our handle on and think about collisions or not as a mechanism. I'll add right now that Bruce Magan also observed that UV irradiation appeared to activate these exact same pathways, and I'll close with that idea for Colin's section. So with that in mind, Colin started exploring these pathways in mammalian cells. The first experiment that he did is either took cells that were untreated or treated with anisomycin, this elongation inhibitor, and he observed potent activation of P38, the MAP kinase. He then asked, if it's ribosomes, do they have to be moving ribosomes in order to activate this pathway? And he asked that question by putting in a drug, herringtonin, that traps ribosomes at start sites and allows all the remaining ribosomes to run off. And he then asked if pretreatment with herringtonin blocked the response, and indeed it does. If you pretreat with herringtonin, you no longer activate P38, meaning that elongating ribosomes, actively elongating ribosomes, are required to activate the pathway. So this was all sounding very good and very ribosome-centric. So then we were ready to ask this key question. If it's ribosomes moving, is it simply stuck ribosomes that have bound the drug? Or is it ribosomes that are colliding that are activating this response? And to ask this question, we used a simple trick that had been developed by Hanny Zayer, and that was the following. If you take cells that are not treated with drug, most of the ribosomes are not colliding. If you treat cells with a high dose of an elongation inhibitor, all the ribosomes will bind the inhibitor and stop. But if you treat with an intermediate dose of drug, the occasional ribosome will be inhibited and the others will collide. So that was the idea. What's the evidence that that actually works? Well, what we can do is we can take these lysates from these three different treatments. We can digest all the free messenger RNA as though we're making a ribosome profiling library. And we can ask whether we generate mostly monosomes, and we do. So these are fractionated over a sucrose gradient. These are ADS ribosomes and disomes, and there aren't very many. And if we do the same treatment with intermediate doses of drug, what you see is that there's in fact a much bigger peak of disomes, consistent with the idea that we've caused collisions at this concentration of drug. So that's our biochemical correlate. This is the method we can use to induce collisions. With that trick in hand, Colin went on to ask whether it's single ribosomes or colliding ribosomes that preferentially activate these pathways. And the experiment's quite clear and simple. If you titrate anisomycin, you can see at high doses of drug, you don't activate these pathways, neither P38 nor junk. But at intermediate doses, it's a very potent activation. Colin went on to discover that another stress response pathway, the integrated stress response, at the heart of which is EIF2 alpha phosphorylation, is similarly activated at intermediate, but not high doses of drug. So from that, we were able to start thinking about how these pathways were activated directly by the ribosomes. So at the top is stress, the stress is collisions, and the collisions lead to the activation of P38 stress response pathways. So for those who study these pathways, and as I learned, at the top of these pathways, above the MAPKs are MAP2Ks and MAP3Ks. So we thought we should try to identify which MAP3K might be important for this activation. Colin did a CRISPR screen asking how cells survived on anisomycin in order to identify factors that were critical. And in that screen, he identified a MAP3K known as ZAC. It had previously been implicated in these pathways, so that was disappointing and reassuring. And with that idea in mind, we went on to make CRISPR knockout cell lines with Sergi Rougeau's lab. So there's a ZAC, those are cells that have ZAC. These are cells lacking ZAC. And you can see that the activation of p38 is completely abrogated in cells lacking ZAC, consistent with the idea that it is the upstream activator. In data I won't show you, we also showed that ZAC was upstream of GCN2 kinase as the key kinase that phosphorylates EIF2 alpha. There are three other kinases in this family, and GCN2 is the critical one. So that was a great start to understanding what was happening. We have a typical MAP kinase signaling pathway at the top of which is ZAC. If it's a typical MAP kinase signaling pathway, we would predict ZAC would be phosphorylated itself in response to collisions. So we went on and asked that question. This data should look now familiar. No drug, high drug, no activation of p38, intermediate, great activation. Colin went on to ask if ZAC itself is phosphorylated. Shown here is a phos-tag gel where ZAC's migration is retarded by the phosphorylation. And you can see that this happens at intermediate, but not high doses of drug, consistent with the idea that as a result of collisions, ZAC is phosphorylated to activate these pathways. This was all looking quite consistent with views on these pathways. The next question we asked was, okay, if this is at the top of the pathway, does it actually interact with ribosomes directly? So we did a simple experiment, or Colin did a simple experiment when he went on and asked if ZAC associates with elongating ribosomes. He did that by fractionating lysates across sucrose gradients under no drug, medium drug, high drug, and running Westerns across these blots. And what you can see is that ZAC, in fact, does associate with the elongating ribosomes, at least it's out there. And it's very substantially out there, I should say. But it being out there doesn't depend on the drug treatment, suggesting that ZAC is just constantly monitoring, but not specifically recognizing just collisions. So at first this was disappointing, but then we realized we should go on and ask whether ZAC itself is phosphorylated specifically on collisions. And to do that, Colin digested these with the RNase again to look at monosomes and disomes. And those data are shown here. This is a digested lysate. So this is now a single ribosome, and this is the disome peak. And looking at ZAC across the gradient, what you can see is that unphosphorylated ZAC associates a little bit across the whole gradient. But that phosphorylated ZAC, the activated form of ZAC, is primarily in this very small disome peak, consistent with the idea that ZAC is associated with colliding ribosomes and it becomes activated. So we've done a lot of other biochemistry that I can discuss, but for the moment, that's what I want to share about the biochemistry. We don't have a structure. We're working on getting a structure with Roland Beckman's lab, and I'm happy to take questions on that. The other thing I want to share with you about Colin's work, however, is what happens. So I should say these drugs are great. We learned a lot about mechanism, but they aren't very physiological. And so we remembered at this stage that UV irradiation was also a trigger of these pathways. And so we went on to test this. Colin was able to show that UV irradiation treatment of cells essentially behaved identically to anisomycin treatment of cells. The same pathways were activated and in a ZAC-dependent manner. So it really did look like another version of the ribotoxic stress response. But why then does UV light give you a ribosome phenotype? We began to think about what we all think about when we think about UV irradiation. We know that UV irradiation causes DNA damage through the formation of pyrimidine dimers. That leads to the recruitment of these kinases, ATR and ATM, when you have double-stranded breaks, and the activation of other MAP3 kinases and p38 activation in J and K. However, we know that if we look at these papers, these responses appear to show up in hours, on the order of 12 hours. Whereas we knew that the ribotoxic stress response that we had been studying happens on the order of 5 to 10 minutes. And so we began wondering how that might be. And we, of course, appreciated that if there's DNA damage with UV stress or UV irradiation, there may well be RNA damage in the formation of pyrimidine dimers, which could lead similarly to ribosome collisions. And that turns out to be the case. We did 3 different types of experiments. They should all be familiar now. We did a polysome profiling experiment to ask if there was the accumulation of collisions. You've seen this before, here it is again. Untreated cells, very few disomes, UV treated cells, lots of disomes. Colin did a ribosome profiling experiment to ask where the collisions were taking place, on which codons. And this is just a quick glimpse, which shows you that, for example, there are collisions happening if you do a metagene analysis and align all the reads at AUU codons, which have a pyrimidine dimer in them. And you can see in untreated cells, there's not a slowdown there. Whereas in UV irradiated treated cells, there's a huge slowdown at that codon and a collision behind it. And finally, and I think the most beautiful experiment, what Colin did is a simple experiment to eliminate the possibility that the DNA in the nucleus was at all responsible for what we were documenting. And he did that by taking messenger RNA and either treating with UV light or not, and transfecting directly into mammalian cells such that the nuclei were untouched by UV irradiation. And he beautifully saw that untreated messenger RNA in the left lane doesn't activate ZAC, P38, JNK, EIF2 alpha, and it makes plenty of EGFP because it's an undamaged message, whereas the damaged messenger RNA activated each of these pathways, including EIF2 alpha, and no longer makes protein because the mRNA is in fact damaged, demonstrating in this simple experiment that the mRNA damage is sufficient to activate the ribotoxic stress response. So that puts us at the sort of end of Colin's story. And I'm going to summarize it here with some cartoons. So what Colin found was that ZAC, the MAP3K, is constantly looking at ribosomes, monitoring collisions. And then when collisions occur, it plays a role in binding to the collision. We think first in recruiting GCN2, 1, and 20 to the ribosome that are involved in activating the phosphorylation of EIF2, which results in a general initiation block in the cells that blocks ongoing translation initiation, thus in some way relieving stress in a cell and giving the cell an opportunity to recover. And that's broadly known as the integrated stress response. And it's really a way for the cell to survive. And some of the most beautiful work, I should say, on these pathways was really done in Alan Hinnebusch's lab here at the NIH. What I'm showing here is sort of a little thing that we were thinking about as we were thinking through these models, is that there must be some way in the cell to monitor collisions. When there are very few collisions, you target the message. When there are few more, you activate survival pathways. And failing that, failing a survival pathway working, ZAC gets phosphorylated, probably falls off to activate downstream MAP2Ks and MAP1Ks in an apoptotic response. So the cell has said, we can't fix this problem anymore, and the solution is to do something more drastic. I'm going to highlight right here that JNK stands for June N-terminal kinase, and one of its substrates is June, and June's going to show its head again in the next section. So we were very excited by these ideas that ribosomal collision might be a very general way for the cell to respond to stress, that these collisions are like the canary in the coal mine, which is sort of the initial response of a cell to respond to stress before the DNA damage accumulates and activates DNA repair pathways and other events. So now I'm going to tell you a second story, and then they're going to come together at the end. The second story is the work of another postdoc in the lab, Niladri Sinha. And Niladri was also interested in the biochemistry of collisions, and he was interested in these pathways for mRNA decay and proteolysis. And he was interested in identifying new factors that might be critical to these pathways. And so Niladri did the same experiment that you've seen already. Niladri took untreated cells treated with a high dose of drug. He used emetine instead of anisomycin, and cells treated with an intermediate dose of drug in order to genome-wide trigger collisions. What Niladri wanted to do, however, was start with sort of an unbiased approach to say what factors bind to colliding ribosomes. Can I identify them in an unbiased manner? So what Niladri did is he either took untreated cells, shown here as UT, untreated, and EL are the emetrine treated cells, and he fractionated these treated cells across sucrose gradients. And what he was interested in knowing is what accumulates in the orange traces relative to the blue traces. So what are the factors that accumulate deep in the polysomes with the colliding ribosomes relative to the untreated cells? And he did that using mass spec. We collaborated with Wade Harper's lab at Harvard. We did TMT mass spec analysis of all the gradients across 11 fractions across these gradients. And then we asked what's enriched in the orange relative to the blue fractions. What I'm going to show you is just one example, which is fraction 8. I'm going to show you what those data look like. Those data are shown in a volcano plot where the x-axis shows you the enrichment of factors in the treated samples over the untreated samples, and on the y-axis, the significance. You can see from these highlighted dots, there are lots of things that appear to be enriched on colliding ribosomes, and a lot of them are interesting to us. But there clearly was an outlier that seemed the most interesting, and it's shown here. It's called EDF1, which stands for endothelial differentiation factor 1, which was known to be a transcriptional coactivator, important for endothelial differentiation. We were really excited about this factor for a week or two because we thought it hadn't been seen before, and then we looked more carefully, and it in fact had previously been implicated in yeast in quality control pathways under the name of MBF1. Nevertheless, this was a super exciting candidate to follow, and so Naladri went about figuring out what it might do. You might be curious, does this show up just in fraction 8, or is it enriched across these deep fractions? And what you can see is it's very enriched from fractions 5 to 10 relative to untreated cells, but in 40s, 60s, and 80s fractions from those polysomes, it's not enriched at all, consistent with the idea it's enriched in deep polysomes. I love a Western. I love to see it with my eyes. And so we ran a Western. Naladri ran a Western with untreated cells, low dose treated cells, or high dose treated cells. And then he looked across the fractions of the gradient to see where EDF1 sediments. And in this lovely experiment, what you can see is that on untreated cells, EDF1 sits at the top of the gradient, not associating with ribosomes, and also in high dose treated cells. Whereas in low dose drug treatment, EDF1 is fully recruited to these elongating ribosomes, and we thought likely to colliding ribosomes. So that was quite reassuring validation of the mass spec studies. Naladri then went on to ask a number of questions, and I'll just quickly go through some of the things that he learned. He first asked, where does the EDF1 bind? And for that experiment, we collaborated with Roland Beckman's lab in Munich and his student Katrina Best. And it turns out they were able to dig in some samples. They had solved the original structures of colliding ribosomes, and they had developed a system to look at trisomes, colliding trisomes on a particular messenger RNA they were studying. And in fact, when they looked hard at those structures, they actually were able to see density for our factor, MBF1. These are yeast ribosomes, so it's the EDF1 homolog. What you can see is MBF1 binds in the second and third ribosomes of the collision, but not in the lead ribosome. That was interesting to us. How does this protein sense that it's in the lagging, not the leading ribosome, when in fact the binding site looks fully intact here? And I'll show you at the end a mutant that we've studied, which is actually missing from this, I should say, missing from the cryo-EM structure is 23 amino acids that we think might be the thing that senses the neighboring ribosome and recruits this factor only to the colliding ribosome. And I'll show you evidence of that later. So this was an exciting structure. It bound in a beautiful place to explain a number of the phenotypes Beck-Grahak had followed. It was only in the collisions. It was a very reassuring structure, and I'm not going to tell you more. Naladri also went on to ask what EDF1 does, and he did what a geneticist does, which is he knocked out the factor and asked what sorts of things don't happen anymore when you knock out the factor. And I'm just going to tell you about 2 of them, and mostly about 1. But I'll tell you the first thing that Naladri did is that it was super obvious, which is to repeat the mass spec experiment, now in a wild type strain or in a strain lacking EDF1. And we took fewer fractions this time. And he simply wanted to ask, are there proteins that are recruited to colliding ribosomes in a wild type cell that are no longer recruited in a mutant cell that therefore depend on EDF1 for their recruitment? So it was a simple idea. It yields another volcano plot. And in this case, we were interested in things that were enriched in this left quadrant, because these are the things that depend on EDF1 for their recruitment. And suffice it to say that we found a number of interesting factors, and I'll tell you at the end what these do. These end up being quite an interesting set of factors that are involved in translational repression, which Naladri validated using reporter assays. He validated that they were playing a role that was EDF1 dependent. But the experiment I really want you to focus on is this one. Naladri then went on to ask, okay, I want to know what happens in cells when there are collisions, generally speaking. And so he did a simple RNA-seq experiment in cells that were untreated or treated with low dose of drug to induce collisions for 30 and 120 minutes. He made RNA-seq libraries and he asked what genes are upregulated or downregulated. And then he repeated the experiment in the EDF1 deletion strain. First, I'm going to show you what happens in just wild-type cells. It's another volcano plot. And what you can see is the collection of genes that are upregulated to the right, significantly on the y-axis, under conditions where collisions are induced for 120 minutes. And what you can see is a number of genes. And if you were a transcription person, you probably would look at this and say, this looks kind of like Jun activation. And in fact, at the top of the beautiful list of candidates is indeed Jun. So Jun is upregulated and it's transcriptionally upregulated and it's quite significant. What I can tell you is that when we ask whether these factors are activated in the EDF1 deletion line, we find that they are not. And I'll just show you that data for Jun. These are wild type cells in black, untreated, 30 minutes, 120. This is the significant upregulation of Jun that we observed. And you can see that that upregulation is significantly lost in the EDF1 deletion strain. And I can tell you it's true for essentially all of those transcripts that were upregulated. So what we're seeing is an EDF1 dependent transcriptional program being activated. And at the heart of that program is Jun. So with that, I'm going to summarize Naladri's story and tell you what we found. What Naladri found was a factor that's recruited to colliding ribosomes. Among the things that it does is it recruits factors, GGF2 and 4E2, that he was able to show block initiation to prevent ongoing translation on problematic sequences, which sounds a lot like a survival choice. And failing that EDF1 is critical for activating a Jun-dependent transcriptional program that has the ring of apoptosis. So you should be a little bit irritated with me now because I've told you two stories as though they're completely independent from two postdocs doing two beautiful stories. And what I'm going to do now is tell you how we're thinking about them together. So I'm going to go back to cartoons because we don't have structures for Zach. But what I've told you so far is that we've identified two factors, EDF1 and ZAC, both of which appear to be critically important in being recruited to colliding ribosomes to activate cell fate-like decisions. I've shown you from Collins' data that ZAC activates this downstream kinase, Jun N-terminal kinase. And I've shown you that there's a Jun-dependent transcriptional response in the nucleus that's somehow dependent on EDF1. And so this all looks kind of nice and like you can kind of fit it together, but I haven't told you one last critical fact. And that's that about 20 years ago, there are a number of studies, one from Japan by Hiroshi's group and one from Kevin Struhl, showing that in the cytoplasm, EDF1 actually associates with Jun. So they associate by co-IPing. That EDF1 is actually a transcriptional coactivator that goes into the nucleus. I'll tell you to begin that EDF1 in general in cells, we think is found in great excess over Jun, maybe at a ratio of 40:1. But what looking at that is allowing us to do is to think about how these things might fit together. And the way we're thinking about it is that this is sort of a two-hit model for activation of the ribotoxic stress response. You don't want it to be true that every time there's a little collision in the cell gets all twitchy and activates this pathway and turns on signaling when collisions have to be relatively abundant. And so the idea is that what the cell does is it activates ZAC, it pulls in EDF1, but it's not until it pulls in all the free EDF1 and then this EDF1 that's complexed with Jun that then releases Jun in order for it to then be able to be activated by J and K, which has been activated by ZAC. And so it's a way to ensure that the program isn't overactivated. So that's our working model. And I'm checking my time. I think I'm fine. So that's our working model for what's going on. And we're trying to think of ways to test it. There's a lot we don't know. We don't know what the structure of this complex might be. We don't know what the structure of ZAC is on the ribosome. We know that Jun is actively degraded by E3 ligases constantly in the cell, and we don't know how to fit that in. But we have been able to ask a few specific questions. I should say the first thing we've done is we've switched to a different cell line. So most of the work we've discussed so far was done in HEC293 and other sort of lab type cell lines. We've switched to a cell line Hackett's, which are keratinocytes, which naturally respond to UV light. This is for the ribosome people in the audience, because these are the nicest polysomes I've ever seen. So these are cells whose business is making protein, I think mainly keratin. We've shown that in these cells, these responses are retained. So these are untreated cells on the left. You can see in UV treated cells, we see clear activation of Jun. We see clear phosphorylation of eIF2 alpha. And we see evidence of ubiquitination of ribosomal protein S10, which is a sign of collisions happening. So these cells are behaving as we would expect them to, and we've started to take apart these pathways there in a little more detail, mostly focusing on EDF1. So the first experiment I'm going to show you here is the model suggests that EDF1 is a negative regulator of Jun. EDF1 is keeping Jun under control and not releasing it for activation. So the prediction then is if we deplete EDF1, we should constitutively activate Jun, and it should be available for phosphorylation and activation of this transcription response. I'm showing you just a simple experiment here where we have untreated or UV treated cells, and we've either depleted ZAC to show the response is ZAC dependent, but more importantly, we've depleted EDF1 with an siRNA. What you can see is in untreated cells, even there, depleting EDF1 leads to some constitutive activation of Jun, suggesting it's been released and is available to be activated by whatever constitutive activity of J and K there is. And we can see in UV treated cells that we really hyperactivate Jun in the absence of EDF1, consistent with the idea that it's functioning as a negative regulator. So that's a little piece that fits in. A second question we've asked is, okay, if these two are together and collisions induces the recruitment of EDF1 to the ribosome and releases Jun, we should be able to do an IP experiment where we IP EDF1 and we ask for a look at association of Jun as a function of collisions. Those data are shown here. This is a strep tag version of EDF1 where we pull down on Jun and get a lot. And when we treat cells with emetine or with UV light, both of which cause collisions, we see the loss of association of Jun with EDF1, consistent with the idea that this complex is regulated by ribosome collisions. And finally, we have an experiment where we have a mutant of EDF1 that we've shown is actually no longer effectively recruited to ribosomes. And I mentioned this when I showed you the structure. Naladri deleted the 23 amino acids at the N terminus of EDF1, which blocks its recruitment to ribosomes. And what he finds is that when he blocks EDF1 ability to be recruited to ribosomes, we no longer see the Jun is released in order to be activated. And so those data are shown here in wild type cells with a wild type EDF1. We see activation of Jun when we expose them to emetine. But when we delete that N-terminal amino, those 23 amino acids, activation no longer takes place. So these ideas are consistent with the idea that, or these observations with the idea that EDF1 must be recruited to the ribosome in order to release Jun for activation. So this is by no means the whole story, but this is our working model. And I think it beautifully explains how two different factors might in fact be important. Their recruitment to the ribosomes might be critical, and how it keeps the system from being hyperactivated and sort of too ready to respond. And it waits for a really significant problem until EDF1 is fully recruited in order to turn on these cell fate decision pathways. I told you about the collision meter, and the way we're thinking about it now is that in fact, this is the collision meter. So the collisions happen, they get sensed, but it's this final release of Jun that really is the threshold setter for how the cell should respond. So those are the data and the story I'm going to tell. I'm going to close with just two broad slides to sort of step back a little bit. We've learned a lot. You know, Colin has taken me head first into this area of cell signaling pathways, and there's a lot I don't know. But I think, you know, certainly we can all appreciate that MAP kinase are very general signaling pathways. I've heard about Jun my whole scientific life, and I know it does lots of things when phosphorylated, involving lots and lots of different outcomes. And there are lots of inputs into the MAP kinase signaling pathways, and they certainly aren't, you know, ribosomes are certainly not the only pathway. But I think these earlier studies by Bruce Magan suggested that there was a ribotoxic stress response, and it mostly focused on the fact that ribotoxins could trigger it. So things like ricin and anisomycin and cyclohexamide. and other natural ribotoxins. But I think what we're learning is that these ribotoxins, additional ribotoxins, there are many general stresses in the cell that actually lead to a ribosome distress phenotype, which should bring you back to thinking about that first slide I showed you, where the ribosome really is such a dominant component of the cytoplasm that it makes sense that the ribosomes could be the canary in the coal mine. And just to give you a sense for how broad that is, what's shown here are some of the data that Naladri's recently collected, and it kind of reiterates what's in the literature. Untreated cells don't activate Jun or junk, but in addition to sort of classic ribotoxins, we found that UV light, doxorubicin, and 4-NQO, which is an oxidative stressor, all activate junk and Jun in a ZAC-dependent manner, consistent with the idea that these are ribotoxic stresses. Whereas cytokines like TNF alpha do activate Jun, not through junk and not through ZAC. So these pathways are diverse, but that this ribostress pathway is much broader than we maybe previously thought. And moving forward, among the things that we're thinking about in addition to these sort of biochemical aspects, is we're trying to think about how different cells might differently balance their need to direct messenger RNAs for quality control, the survival, and the apoptotic pathway. And you can imagine these would be regulated by the relative abundance of the factors involved in these pathways versus these pathways, or by the ratio of EDF1 to Jun, and then it might be different in neurons and plasma cells. And so those are some of the approaches that we're beginning to take. So with that, I'd like to thank again those who did the bulk of the work here. Colin Wu was an amazing postdoc that I truly miss, and I'm so happy that the NCI has him there, starting his own lab. Naladri Sinha is a wonderful postdoc still in the lab, and this is Jake, that was his rotation project, was to work with Naladri on this great project, and you can see he was very happy to do so. This is my lab at a COVID lunch, but let me acknowledge other people here. Sergi Rougeau is my colleague downstairs, and his student Amy Peterson helped with the ZAC work, and Sergi studies MAP kinases in live imaging and has been incredibly helpful in helping us to think about this. The mass spec was a collaboration with Wade Harper's lab at Harvard and Eric Bennett at San Diego and his postdoc, Alban Ordero. The cryo-EM was a collaboration with Roland Beckman's lab in Munich and his student, Katrina Best. Thank HHMI and the NIH for funding, and I'd love to take any questions.
Rachel, thank you for that beautiful talk. I do want to mention one thing real quickly. And that is we're lucky that Dr. Nurnberg's wife, Dr. Myrna Weissman, was able to join us. And she knows a bit about ribosome profiling, or has heard about ribosome.
I'm guessing.
So anyway, I'm going to go straight to questions. To one question that I also had has come up a couple times, and that, how is ZAC phosphorylated? Autophosphorylation, or what is it sensing to get for it to be phosphorylated?
Yeah, that's a great question. I mean, we don't know anything structurally, but Colin did do a lovely experiment where he actually had he had cells where he had endogenous ZAC present and a trans version, whatever, he put in an extra copy of it that was either kinase dead or kinase alive. And through that experiment, he was able to unambiguously show that it was a cis reaction. So it's not a trans phosphorylation reaction, but it's a cis reaction. So we assume it functions as a monomer. Yeah, it was a very clear experiment.
Okay. I need to do one piece of housekeeping. The CME code for everyone is 31297. Let me go back to questions. EEF1A has been involved in the degradation of stuck RNAs and uncompleted polypeptides from translation elongation. Has EEF1A, does it have any involvement in ZAC phosphorylation? This is from Juan Marugan.
Yeah, that's funny. I saw that paper for the first time this week. I think it's Nam Sonnenberg's paper with Ivan Topasorovic. I have no idea how EEF1 might be implicated in this pathway. We haven't explored it. You know, I always think of EEF1A as the most abundant protein in the cell. And so when it comes up, I tend to ignore it because it's so abundant. But that doesn't mean it's not real or true. So I don't know the answer to that question.
Again, this is from Poonem. Great talk. Does EDF1 affect ZAC phosphorylation or binding to ribosomes?
Yes. We don't fully have that answer, but we are working on that answer. What we do know is that EDF1, an EDF1 delete, there's less P38 activation. But it's a modest effect. It's not a full effect. And so when we published that paper with EDF1, our interpretation was that EDF1 is really good at stabilizing that interface and making it tight, and that maybe ZAC would bind more loosely. Naladri's been working at getting ZAC to see if ZAC is phosphorylated in that situation, and we don't have that answer yet. But my bet is it's gonna still be mostly phosphorylated.
Next, I have a couple questions from Vula Millie. The first question is, what happens to the ribosomes involved in collisions during stress recovery?
Yeah, that's a great question. You know,
can they be recycled and reused?
Yeah. I mean, it certainly would seem extremely wasteful of the cell to dump the ribosomes who had it was a no-fault accident, right? They weren't at fault in the problem. And so yeah, I spoke with, I think, Dan this morning. You know, my bias is that ribosomes can get stuck a lot, and mostly they get off and they get reused, and we don't have any evidence to the contrary. There's a pathway known as non-functional ribosome decay, and you know, where a ribosome is at fault. And I imagine the way it happens is the ribosome gets a ding every time it makes a mistake, and eventually the dings, it could be a ubiquitin or something, that signals that the ribosome must have been at fault if he did it enough. But I don't know that. I know I'm not supposed to anthropomorphize, am
I? Well, do dinged ribosomes work less well related to that? And then Vula has another question.
Yeah, I think we have no evidence. You know, we don't have any evidence. You know, ribosomes do get ubiquitinated as a consequence of the collisions. And we think the role of that is to recruit factors for their rescue and for these other quality control events. So I assume all of those things happen. And I think we all assume that the ubiquitin is then removed. There are dubs that are associated with the process that probably remove them.
Yeah, so that was basically you're answering Vula's second question was, are the ubiquitinated R proteins removed or affect subsequent rounds of translation?
Yeah, to be honest, I think I've answered it, but to be honest, we're very interested in that question because we wonder if they aren't removed.
Okay.
But we don't know.
Alan Hinnebusch has the following question. EDF1 is an inhibitor of Jun in your model, but was originally identified as a co-activator, same as MBF1 for GCN4. How can this be reconciled?
Yeah, this is a great, I could count on Alan to get me that question, but I did my homework. I was anticipating this. You know, we can't reconcile everything. So I'm proposing it plays a negative regulatory role on Jun, and yet I showed you data where it was functioning positively in transcription. And we assume that the transcriptional data that we reported in the published paper is the result of that role as the co-activator. And we're going to do CHIP and see if it's there. We do have data looking at cytoplasmic to nuclear fractionation, and we see EDF1 does go into the nucleus after collisions occur. And we don't understand how to sort out the fact that it goes to ribosomes as well. So we see it go to ribosomes and we see it go to the nucleus, and we don't know which populations are doing what. We are very confused. That's what I told you. I wanted you to pay attention, Alan, and figure that out for me.
So maybe Rachel, on that note, I want to thank you again very much for such a lovely talk that covered so many different topics. And thank everyone else who has been listening to this.